something hanging down, but I couldn't see the bottom of her leg. I'm like, Dr. Reynolds wearing a dress? Surely not. Just, yeah.
I suppose it could be. It'd be a little short in the front.
Everybody's draping.
It's a very long sweater. How are you guys today? Ready to learn? I'm super excited. I'm pumped. Today we get to draw. Awesome.
We are artistic.
We are going to draw.
Drawing is not awesome. Run away from drawing.
Drawing is fantastic. It's a diagram of science.
Well, yeah, it's just like a chemical cell, but this is a regular cell.
Pretend that it's a giant cell that encompasses the Konza pairing. All right. So today, oh, you know what? I bet we better talk about our prairie critters.
You want to do plants? You want to do animals? You want to do both?
I would rather not do the plants.
Okay.
Because I haven't looked at what they are.
Well, I haven't either.
I know, but you've done this before.
Of course, I didn't used to do the plants.
Oh, that's true.
Wow, I wish I could see.
Both? I would like. Hang on, we're gonna zoom through here.
Check your foot.
Let me find where we're at here. I forgot to do it last time.
Okay, so then we did last time I was here.
Yep, that's it right there.
Ah, switchgrass. Switchgrass, Panicum virgatum. It's C4 grass, so it's in the same class as Indian grass and big blue stem. You've already learned now about C4 photosynthesis, so you all know exactly what I mean when I talk about C4 photosynthesis. It's another one of these grasses that is well known for its extensive root system. It also reproduces due to rhizomes. It is one of the species that people are exploring as one of the options for producing sustainable biofuels. It grows quickly, it grows in dense clumps, and it can be harvested and digested to create biodiesel, essentially. So, Panicum virgatum, you'll see, I don't know, there are probably 3 or 4 universities around the country that have switchgrass programs that are exploring this for fuel.
The University of Illinois has one of the largest grants that NSF gives for switchgrass.
Switchgrass. The nice thing about it is drought resistant, so you can grow it in places where you can't grow other things. And right now, most of our biofuels are produced by corn, which requires huge inputs of nutrient and phosphorus fertilizers, is subject to perennial pests, corn rootworm, and things like that. So this would be, if you can get it to work, a viable alternative. Dahlia purpurea, prairie purple clover. This is a legume. It's in the family Fabaceae marker.
Oh,
Markers.
sorry. In this pocket.
I did not bring my marker.
Expect to be lecturing or the day that I was supposed to lecture?
Yeah, so things in the family Fabaceae, remember that plants are different. So Fabaceae, the CEAE suffix is for plant families. This is the family of the legumes. The legumes have pinnately compound leaves. They're leaflets with lots of little bitty flowers, or little bitty leaflets on them. They produce closed flowers. The peas are in this family. These are also the ones that have root nodules associated with the roots, outgrowths of the roots, where they support nitrogen-fixing bacteria. So these are nitrogen fixers, are all in this family. It's a perennial herb. It has a deep taproot. That's how it makes it in places like Con's Prairie. It's commonly used, those of you doing prairie restoration work or work on restored prairies, commonly found because it has a nice, pretty flower, and also traditionally used in traditional medicines by Native Americans.
Do you know for what?
I don't know for what. I should
Either.
look.
I should look up.
Da, 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 da.
Wild spinach was used by Native Americans as an antibiotic, but it does not grow on the prairie.
It's edible. It's good for making teas, and it says here that. Yeah, I'm not sure.
Okay.
I'll maybe look it up.
Edible, apparently.
Yeah.
Just you eat it.
Well, actually, it turns out lots of legumes are edible. The prairie animal of the day is the bull snake, Pituophis melanoleucus. Actually, I think the taxonomy has changed recently on this, but we'll just call it melanoleucus for the moment. It's called melanoleucus because it's dark on the front end and then gets lighter as you go to the back. People oftentimes confuse this with a rattlesnake because it tends to hiss and also wiggle its tail in leaf litter. It is a super common snake on the prairie, but you don't see it very often because it's usually down in rodent burrows feeding on rodents. We occasionally catch them in the field when we're out with students. This was on a tri-beta trip years ago. They can lay quite a few eggs, 3 to 24 eggs in the early part of the summer, and they can become quite long. It's one of the larger North American snakes. This is kind of a medium-sized specimen that we caught out at the Tallgrass Prairie Reserve. Once you get them tamed, they're actually quite delightful. People prefer them as pets. It's a wide-ranging species. Goes all the way from the eastern seaboard all the way over into the Midwest. But the Midwest is probably where it thrives the best. And those are your prairie plants and animals of the day. And here's your murder back.
Thank you.
You want lights back on?
Are those
Nah.
all the things that they available and everything? Is that going to be on the tests?
Yes, they will be on the test. And you just have to know about their general biology, just what we've talked about in class. It's important for you to not forget that the things that we're talking about are actually organisms, and so we want to kind of familiarize yourself with some actual species rather than just thinking of ecosystems. Somebody else have any hand up? All right. That's the reason we're doing it, is to refocus us in this place where we're talking really about things that's really broad scale, that the things that are driving all this are species.
Right. Makes a lot more interesting when you think about the day-to-day life for me of the critters in that area. All right, so we are going to continue our discussion of greenhouse gases, and we're going to go into nitrogen cycling instead of carbon cycling. We're talking about carbon cycling on Friday. We're going to go into nitrogen cycling. As a reminder, carbon cycle, a cycle is a collection of major pools and fluxes, right? So what were some of the carbon pools that we talked about on Friday, for example? Was it the ocean? An ocean, yeah, the ocean was definitely a pool of carbon, absolutely. What's another pool of carbon? The plants themselves, absolutely. Yep. Another pool of carbon.
Earth's crust.
The Earth's crust. Mm-hmm. Yep. What about some? Those are not all of the pool. Oh, go ahead.
The atmosphere.
The atmosphere. The atmosphere is a major pool of carbon. What about? Those are not the only pools, but what about some fluxes of carbon? Some flows of carbon from one direction to another, from one pool to another. What are some things that cause a flux of carbon?
Photosynthesis.
Photosynthesis, sure, yeah, absolutely. That's the major carbon flux that we've been talking about thus far, right? Taking CO2 from the atmosphere, fixing it into plant biomass, putting it into that plant pool. All right, what's another flux of carbon? Caleb, what's a flux of carbon going from one pool to another? The atmosphere to the Earth. Okay, cool. And what would cause carbon in the atmosphere to go to carbon in the Earth? Okay, plants, we talked about photosynthesis. Okay, so that gets it to the plant. How does it get to then to the Earth?
No idea.
All right, let's keep thinking.
Rain?
Well, rain isn't going to move it from the plant to the Earth. You might have some carbon particulates in the air, though, that would be caught by rain and go to the Earth, yeah? What would put carbon particulates into the air? What's another way that carbon can go into the air?
Cows.
Cows can fart methane, that's true. Absolutely, there's lots of cow farting going on.
Through respiration.
Through respiration. So cellular respiration can break down, right? It breaks down these carbon compounds and then we breathe out CO2. Absolutely. Okay, and that can go back into the atmosphere. Sure. All right, so be sure that before the exam you go back over that diagram of all the pools and the fluxes, and so that you can come up with ways, when asked, that carbon can go from pool to pool and identify things that are pools versus fluxes. Okay. Well, just like the carbon cycle, having these pools and fluxes we talked about. Here's that diagram, right? And we ended last time talking about atmospheric CO2 concentrations. Historic CO2 concentrations can be obtained by sampling ice cores from icebergs, basically. Okay. So we've got basically sampling from the actual atmosphere from whenever humans started being able to do that. Before that time, we can sample ice cores and find out what the CO2, atmospheric CO2 was like based on the CO2 concentration within the ice core. Here's the year 2000, here's the year 1900, so you can see this goes back quite a ways, 2,000 years. And you can see the increase in CO2 going up drastically after the onset of industrialization.
Derek Thurman will be talking about this afternoon in his primary literature paper in undergraduate research, and he has a NASA data set that goes back 400,000 years.
That's fantastic.
It's all from ice cores, and it shows really big fluctuations, but nothing like what's going on today.
Right. Another thing that you guys will see when this kind of thing comes up, people say, well, you know, CO2 and temperature, they've fluctuated over time as the Earth goes through natural cycles of cooling and warming. And that's true. You'll see, oh, that's not good anymore.
Cool.
It caught.
Luckily, somebody who sucks as bad as I do at throwing markers away. I never hit the basket.
I can usually get really close, but I got caught up on the costume here. All right. So you'll see them saying, okay, well, there are natural fluctuations, and that's true. You know, you'll see things go up and down like that. You know, but then you plot this, and it's not like anything that we've seen in the past.
There's a dip there, and I can't quite tell what year that's at. Where is that?
1600.
1600?
Mm-hmm.
That happened in 1600. That caused a massive dip. It's not the plague.
Right here?
Or that. Genghis Khan.
My guess is that it's something to do with a combination of volcanic eruptions and the Little Ice Age. Would that be? He's not paying attention, but Right? So I will, why don't you look into it for us? Not right now. Tell us next time. All right, so why does CO2 matter? Why does CO2 in the atmosphere matter? Well, the only reason that we can support life here on Earth is that we have an atmosphere. Okay? And the presence of an atmosphere, the depth of that atmosphere, the composition of that atmosphere depend on a lot of different factors. But one of the factors is that we can be warm enough to live, right? The atmosphere on Mars is quite thin, and so it's really, really cold on Mars. So cold that we can't live there, not without significant planning and intentional colonization. Here we have this nice fat atmosphere. And what happens is that the sun's rays come down, and these photons, some of the photons get through the atmosphere and are absorbed by the surface, absorbed by plants, absorbed by simply making rocks warm or water warm, okay? Some of the sun gets through the atmosphere and is reflected from the Earth's surface back toward the atmosphere, and sometimes it escapes. Some of that light gets reflected off of the atmosphere and never gets to the Earth. The reason that we're warm is that some of this light is absorbed by the surface, and some of it is then radiates from the surface, is released and is kind of reabsorbed by greenhouse gases in the atmosphere, right? That warmth is kind of, it's almost like steam around the Earth. The bad thing is that if we get too much concentration of these greenhouse gases, then fewer and fewer, less and less of this long wave radiation that is re-emitted from the Earth escapes the atmosphere. And so it'll cause increased warming. The greenhouse gases that have pretty big effects, carbon dioxide, obviously, methane. What's N2O? That's not nitrous oxide, that's some sort of,
Oxide.
pardon?
Nitrous oxide.
Is it nitrous oxide? I second guessed myself. Water vapor and ozone. And we talk a lot about CO2 because that's one thing that we produce a lot of. Methane we produce less of, but has a larger impact on this resorption of heat. It can carry more heat that's re-emitted from the Earth's surface. All right, so this is a depiction of trends in surface temperature. The data are taken from 3 different data sets. The white blocks are basically areas where we don't have data or we don't have high enough quality data. The trends were calculated, the ones that are included are only those that have more than 70% complete records in the first decade of the 1900s and the most recent decade, the one that we're in. You might be able to see these little black pluses. The little black pluses mean that there's consistently an increase in temperature. The color, the shading, indicates the trend in temperature. So if this blue here, that's decrease in 0.4 to 0.6 degrees Celsius. You'll notice these purple areas, that's an increase in something like 2.5 degrees here. So you can see that across the globe, we're getting an increase in average temperature. There are 2 of these because they're just from 2 different sources of data. This is from the International Panel on Climate Change. They meet every year to discuss all of the possible ways of interpreting the data. So when you hear about scientists disagreeing on climate change, they are disagreeing on the extent of how warm it will get and the extent of the change. So they're not disagreeing on whether or not it will increase warming. And when we say global warming, we don't mean that all winters will be warmer. We don't mean that all areas will be warmer at all times. What tends to happen is that average temperature increases, but fluctuations also increase. And so it's basically, I prefer the term climate change because we get these drastic changes. So your winters might actually be even slightly colder. And then summer's much warmer. So that we get an average increase, but we're getting this variation that's much more variable.
What's with Greenland?
What's with Greenland? Well, you probably can't see it. So Greenland's really tiny. It's right here. This is actually ocean.
Really?
Uh-huh.
What's with the ocean south of Greenland?
Right. That's a really good question. I actually don't know the answer to that question. Do you?
Is that a current?
It's a current, for sure. I mean, it's what's like an eddy in a current.
I don't know, it might be due to changes in ocean currents, because they'll shift as well with climate shifting.
That would be my guess.
But I don't know, that's an educated guess.
Yeah.
I just looked at an article from 2015 in Nature. The dip in the 1600s in overall CO2 levels is attributable to the annihilation of people in the New World with the colonization of the New World.
Oh.
Global population declined precipitously, and also all of the farming activity done by those people in the New World went down during that time, and there was less clearing for agriculture as a result. So all of those things. So it was basically, human population decline led to decreases in CO2 level.
Right. So, have you guys read or seen Dan Brown's Inferno? Yeah, he decides that the only way to save the Earth is to get rid of most humans. And as we have seen per the data, that might be a good plan. You never heard it from me. I don't agree with his reasoning, though. His reasoning is not scientifically sound because he uses a different argument. Okay, so we're going to have this increased atmospheric CO2, and yes, it's going to increase more, but, and we have this increased atmospheric CO2, but what is it going to do? So one idea is that plants might be able to cope with the increase in CO2. What do we know about photosynthesis and carbon fixation with increased CO2? It might depend on how much it increases by. What if it increases just a little bit? What happens with carbon fixation? Rate of photosynthesis. Kevin, you know this. What happens to the rate of photosynthesis when you increase carbon dioxide? In the end, you will produce more oxygen, okay, but through what process? Okay, so if you have more carbon in the air to fix, what can you do faster? Photosynthesis. You can fix more carbon, right? And so if we have this dramatic increase in CO2, maybe plants will be able to keep up with this either through acclimation to their environment or through evolutionary adaptation. Okay, so there are several experimental sites around the world asking this question. So one of them is at Duke. This is the Duke FACE site. This is a big forest. And you'll see these little tiny white sticks here. These are giant poles, right? And FACE stands for Free Air Carbon Dioxide Enrichment. Okay? So what they're doing is they've got this big tract of forest here, and they're saying, okay, if we make these artificially increased CO2 plots here, and we can then monitor photosynthetic rate, biomass increase, that kind of thing, so that we can maybe predict what's going to happen, how plants will respond to having such abundant CO2 in the environment. Now, if they've got a ton, the more CO2 in the atmosphere, what's going to happen to the conductance through the stomata? The carbon dioxide through, by diffusion will have a faster rate through those stomata. Okay, so yeah, maybe it'll just kind of suck it up. Here's another face experiment. This is at the University of Illinois. It's their soy face experiment. It's a soybean field, and they have their free air carbon dioxide enrichment. I could only find a picture of one of their plots that was high enough quality. They've got some little sub-experiments going on here. They've made little tents to test a few other things. But they're just sort of releasing CO2 there to see, okay, well, we've got one that's looking at forest growth. Let's look at crop growth as well.
Would C3 plants benefit more from excess CO2?
I think that's an excellent question. In fact, that's going to be part of your homework for Friday, is to figure out how C3 and C4 plants, how you would predict that they would react to increased CO2, and whether or not you think C3 or C4 might do better under those conditions. So that's an excellent question. Okay, so to prep you guys to think about that, we talked about how CO2 diffuses into the leaf from the atmosphere, and how fast it diffuses in is going to depend upon the differential, right, the gradient. If you increase the concentration of CO2 on the outside, then the gradient, the differential between the outside and the inside of the leaf is greater, right? And so it's going to want to flow across that stomata. So you've got increased diffusion into the leaf. That increased CO2 flowing into the leaf means an increased rate of photosynthesis. This is called the CO2 fertilization effect. Okay, so we've got increased atmospheric CO2 means increased diffusion of CO2 into the leaf. Okay, so that's number one. And then number two, this increased CO2 diffusion into the leaf means an increased photosynthetic rate. And this is called the CO2 fertilization effect. What about water loss under this condition? Is this going to change at all the way that plants can be efficient with their water loss or use? Think about it for a second, and then I want you to turn to a partner and talk about how might increased CO2 in the atmosphere change the water use efficiency of plants. Especially given what we just talked about. You guys.
More water. She still was opening up more.
So what we're figuring here is what would be the effect of you have one solution faster,
So, like, they can-
you can introduce some models. I mean, but that's just kind of like things you can always, but because they don't say so. Well, they do more photosynthesis, limiting reactants. In this case, we're yeah, so
There's too much CO2.
So my point is,
To maintain. So
if I were essentially, but then I also excess CO2.
Okay, so what are your thoughts? What are some ideas, guesses? What is this going to do to water use efficiency? Candy?
Well, we talked about basically if the concentration of CO2 is higher than the small body open one, which means there's likely more water loss. So you're gonna have to compensate for that in some way.
Okay, they would be open more. If the CO2 goes more easily into this leaf because the differential is greater, would they need to be open more? That's a good guess, because you want to maximize your photosynthesis, right? And you can get more photosynthesis done, but you actually don't need to have your stomata quite as open to get the same amount of CO2 as you did previously.
Simply because of this simple laws of diffusion. So go back and review your basic chemistry of laws of diffusion.
Right, so if we have our stomata here, and we've got loads of CO2 out here, and normal leaf amount of CO2 over there, you know, those molecules are going to want to even themselves out across the stomata. If we put even more CO2 here, that rate of diffusion is going to be much higher. This is like having a deflated water balloon and making a hole in it, and the water goes bloop out to the other side of the balloon membrane, or having a really inflated water balloon, right? You make a tiny hole and it goes spraying out everywhere, right? That's the same thing as diffusion. And so in this case, although us thinking we're like, oh yeah, I'm gonna open my stomata even more because you know what? I can photosynthesize the crap out of this thing, right? Because I've got loads of carbon dioxide. But that's us not kind of thinking ahead, thinking, okay, well, actually, I could conserve some water and get the same amount of photosynthesis done by closing my stomata a little bit, right? It would be smart and conserve. So, if this is how much CO2 you need on the inside of the leaf, then you can actually close your stomata a little bit and still get the same amount of photosynthesis done, but lose less water. The only reason we know this is because we've actually done the experiment. Because initially it was sort of anyone's guess what would happen. And one of the hypotheses was that you would open your stomata more because you have a better chance at increasing your photosynthesis. But what happens is that we see that plants actually will close their stomata partially because then they can have the same amount of photosynthesis but less water loss. After a while, we start to see them making leaves with fewer stomata and smaller stomata. So their water use efficiency can go way up. Okay, so increased carbon dioxide means enclose stomata partially, or even have fewer stomata. And smaller stomata, and all of this gives higher water use efficiency because they're getting the same amount of photosynthesis done with less water. You guys have questions about that?
Are all of these part of the CO2 fertilization effect, or just the second one?
Just the second one. These are all part of having increased CO2. But I wouldn't necessarily consider, well, I don't know. I mean, I think you could make an argument for number 3 being under number 2, but we can just think of them as separate. They're just all effects of increased atmospheric CO2. All right, so keep these things in mind when you look at that worksheet that I've posted that will be homework for Friday, and you're trying to think about, okay, how is photosynthesis going to change with increased carbon dioxide in C3 plants, in C4 plants, and do I expect a difference in how they'll respond? Okay, so that worksheet is going to be due on Friday in class. Okay. All right, now we're going to transition a little bit from carbon cycles to nitrogen cycles. All right, and one of your goals is going to be to draw a model of a nitrogen cycle at Ponza Prairie. And don't worry, you don't have to be a good artist. It's just a diagram. If you want, you can think of it as a giant cell. Okay. So why in the world would we need nitrogen? Well, there are 4 basic macromolecules that make up all of life. Okay? 4. Only 4. One of them is nucleotides. What do nucleotides make in our cells? DNA. They make DNA, they make RNA, and they make something else that gives us lots of, that we use as energy that I bet you might not have thought about before as being a nucleotide. What's the major energy currency within the cell? ATP.
Oh, that didn't seem like my close.
Right? Isn't it crazy when that clicks? It's like, oh my God. All right, okay, so we've got nucleotides. We've got amino acids. Amino acids make proteins. Most of the jobs in the cell are done by proteins. Almost all of them. Proteins are super crazy important. Rubisco, remember Rubisco that likes to cheat on CO2 with oxygen? Rubisco, that enzyme is a protein. Pep carboxylase, who's super faithful to CO2 and doesn't care about oxygen, that's an enzyme. It is an and some enzymes don't have to be proteins, but it's also a protein. We've got fatty acids. What do those fatty acids make?
Membrane?
Membranes. Yeah, fatty acids make membranes. Membranes, hormones, cholesterol. But the biggest one is membranes. They're what keep us from spilling out into the environment. And then we have monosaccharides. What do they make?
Polysaccharides?
Polysaccharides, right? Which is
Starch.
sugars and starch. And sugars can be used for energy. They can also be used for a label on the cell. A lot of cells will have a little label that says, hey, I'm a liver cell. I belong to you. Don't attack me. All right. Nitrogen is critical, is a huge component of 2 of the 4, and a minor component of the other 2. It is a huge component of nucleotides and amino acids. Amino acids are called amino acids because they have amino groups as a major portion of their structure. An amino group is a chemical group made with nitrogen. Nucleotides all have nitrogenous bases on them. Nitrogenous bases, it's a base, so it's not an acid, it's a mild base, but they're basically these cyclic compounds with a bunch of nitrogens in them. So nitrogen is critical for the majority of the functions of the cell. So plants and animals are pretty reliant upon nitrogen. And just like carbon has pools and fluxes in its cycle, the nitrogen cycle does as well. We're going to think about We're going to think about what those are. Okay. I want you to draw in your notes, or leave a space in your notes so that we can draw the nitrogen cycle at Kanza Prairie. And the types of fluxes and pools that are at Kanza Prairie are going to be very similar, if not the same, to those fluxes and pools present elsewhere. All right, so Kanza Prairie is land. So I'm going to draw soil. Okay, so draw a big box for your soil. And down here is rock. This is the bedrock. All right, we have soil. Okay. Nitrogen often comes in the form of ammonia. What are some pools of nitrogen that you think probably exist in this cycle? What are some of the pools of carbon that we have? Plants, okay, absolutely. So we've got one pool here. We've got plant shoots, so it's like above ground plant matter. And we've got plant roots. Okay, and I just said that pretty much all living things are super reliant upon nitrogen, right? How else could we get, what's another pool of nitrogen in Kanza? Fauna. Okay, yeah, absolutely, animals. And so in Konza Prairie, what's one of the main animals that we're considering in the Venadal research? Bison. Okay, all right. Okay, so we've got plants, shoots, and roots. We've got animals. Okay.
Yes.
The atmosphere, absolutely.
Mostly.
The atmosphere is mostly nitrogen
Useless and
in useless N2. That's exactly right. Our atmosphere is mainly nitrogen gas. That's the most common gas. Okay, so we've got atmosphere. Awesome. Okay, anybody keep a fish tank? All right, what happens when your when you don't clean the tank a lot? If you were to test the water, what would the water be high in? You know? Ammonium, nitrates, okay. Where are those nitrates coming from? Food that's on the bottom of the tank is decomposing, sure. What else?
don't aquatic animals usually excrete ammonia?
Yes, they're pooping, right? Fish basically just poop as they live, right? They're excreting, okay? So we've got urea, okay? So basically pee, okay? All right? If a fish dies in your fish tank, then you really have an ammonia problem, right? Because there's decaying organic matter.
Isn't excretion more of a flux valve?
Yes. So the urea itself is the pool. And I'm gonna add a flux here. So very good thinking. Wasn't gonna add the fluxes yet, but I can add it there. No, Siri, go away. Okay, so we can say
Well, thank you.
excretion,
Especially the nitrogen mixing.
dead, just a second there, organic matter. Yes, microbes. Plants and animals are not the only critters living in the ecosystem. Microbes need nitrogen just as much. Okay. Okay, so we've already talked about two of those fluxes. We can do excretion, we can have death. Some other, we're gonna add some other pools as we discuss. Okay, so you're gonna come back to this diagram and you're gonna add to it as you go along. What about streams? Rivers, rain.
Rivers, they erode the soil, which has nitrogen in it, and they can carry ammonia, urea, that sort of stuff, and then dead matter.
Yeah.
All the stuff that's on land is basically also straight.
Yeah, yeah, absolutely. And so we'll add that flux here in a little bit. The other thing that it does is it makes the groundwater into a pool of nitrogen as well. And as we talk about some of these fluxes, I'm going to add in a few more pools that aren't so obvious until you know about the flux itself. Okay, so fluxes aren't just movement of nitrogen or carbon. They can also be transformation of one form to another. They can be inputs into a pool. They can be outputs from a pool. So for example, groundwater is the result of an output called leaching. It's just water filtering through the soil and leaching nitrogen that's in the soil already into the groundwater. Okay. So fluxes can be inputs, transformations, or outputs. And you can notice from this diagram that it doesn't always have to go directionally from one pool to another. It can go from one pool to another and then back to that other pool, right? So for example, the bison, let's say, is going to go over and it's going to eat plant shoots. And the nitrogen is then going to go from those plant shoots into the animal, and the animal will poop and go into dirt soil, and then the microbes might fix that nitrogen, and then the nitrogen go back into plant roots and plant shoots, for example, right? So we can have cycles, and we can have inputs and exits. All right, so now we're going to think about the fluxes themselves. What do you suppose the main input of nitrogen is? And this says in a pristine system. It just means if humans aren't mucking things up. If there's no humans around, what do you think the main input of nitrogen is into a system? Now, matter is not created or destroyed, so I guess it's not being created, right? So I guess input from the atmosphere, or input into a living system. How does it get from non-usable form to a biologically usable form? How do we do that with carbon? CO2 was kind of our non-usable form. We couldn't use it for carrying energy. So what do we do with it? We fix it, right? So nitrogen fixation is the main input of nitrogen into biological systems and ecosystems. There are some exceptions. You can get water flowing, you can get animals coming in and pooping, but the main input into a system, an ecosystem that's not disturbed by humans, is nitrogen fixation. We talked about how the atmosphere is mostly nitrogen. It's about 80% nitrogen gas. And the form of nitrogen found in the atmosphere is extremely unuseful to us. It's N2, and those nitrogens are sharing electrons. They're covalently bound. Covalent bonds take energy to create. They take energy usually to break. They're pretty strong. Non-covalent bonds are kind of like Velcro. You pull on them, they come apart. Covalent, not so much. Covalent bonds are some of the strongest bonds that you'll find in a biological system. Ionic bonds being stronger, but most of the time we don't have ionic bonds in a biological system. Okay, so we need to get this N2 that's very tightly bound together with this covalent bond. We need to break that apart, and we need to fix that nitrogen into a usable form. And this process is basically taking this N2 and adding 8 hydrogens. Okay, 8 hydrogen protons and 8 electrons. and we get 2 ammonia and some hydrogen. And that doesn't seem like it should be that hard, right? But what you have to do is you have to convince nitrogen to let go of the other nitrogen and to bind with hydrogen instead. Nitrogen is much more electronegative than hydrogen is. And so it's going to hang on to its bonds a lot more tightly than hydrogen would. It's going to be really hard to rip those guys apart. So we have some biological workhorses that can cause these sort of unusual reactions to happen. Rubisco was one of them, right? That fixed carbon dioxide. It said, hey, carbon, I know oxygen is super electronegative and is a giant hog and loves to get all of the electrons, right? But trust me, let go of oxygen, everything's gonna be okay, right? PEP carboxylase did the same thing. They're both enzymes, right? There's an enzyme that does this for nitrogen. Oh. And apparently I'm not going to talk about it yet. My bad. Here, let's go here. Ta-da! It's nitrogenase. Now you'll notice that PEP carboxylase, Rubisco, which is ribulose bisphosphate carboxylase oxygenase, they're ending in ACE, right? They're all ending in ACE. ACE is a signal to you that it's an enzyme that is changing the bonds, the chemical bonds of the molecules that it's interacting with. Rubisco is a carboxylase or oxygenase. It could add a carbon or an oxygen. It could fix a carbon or an oxygen. This is a nitrogenase. It fixes nitrogen. It's going to add a nitrogen. It contains kind of a rare element. It's not super rare, but it's rare as far as being a component of an enzyme that we think about in biology called molybdenum. And I will never be able to say that fast. My apologies. I will always say it molybdenum because I just can't say it fast. It has iron, it has sulfide groups, and it needs ATP as an energy source. It cannot do this reaction on its own. Okay? And that was true of Rubisco and PEP carboxylase too, right? They used ATP and NADPH. Just like that, nitrogenase needs ATP. It requires a lot of energy to break this bond. If you want to convert, if you want to fix 1 gram of nitrogen, and that would be like this much, okay? If you want to fix that much nitrogen, you need 10 grams of glucose and harvest the ATP from that. It requires a ton of energy. So this nitrogen fixation is the process by which we take it from this N2, which is not very useful in the atmosphere, and we convert it to ammonia so that we can use it in our building blocks in our cell. This is done by bacteria, and the bacteria sometimes is in association with plants, like Rhizobium or Frankia. Some of this nitrogen-fixing bacteria is free-living. It's just in the soil, like Azotobacter or Cyanobacteria. It can also just be in the water, like Cyanobacteria. Cyanobacteria is really cool. It's a bacterium that can photosynthesize, which is kind of awesome. I wish I could photosynthesize. I've included here, what do they eat? Most of them are heterotrophic, so most of them eat stuff, right? But cyanobacteria are autotrophic, they photosynthesize. And in the process, they fix nitrogen from the atmosphere or from the water into a biologically usable form. It makes that nitrogen available for plants to make amino acids, nitrogenous bases that will be nucleotides and other nitrogen-containing compounds. And you know where we get all of our nitrogen as humans? Plants. Plus the bacteria that we accidentally eat. Right? We get some from animals, right? But those animals got the nitrogen from the plants, and the plants got the nitrogen from the bacteria. In my molecular genetics class last semester, one of the challenges that the students decided that they wanted to do was to try to figure out how bioterrorists might cause problems and kill others. And one of the ideas that they came up with was to attack nitrogen-fixing bacteria in the location of your enemy. So let's talk about nitrogen-fixing bacteria that are in association with plants. Okay, so this is just part of a root. You can envision it, it's a cross section of a root, so it would be a circle here, and it would have a vascular cylinder in the middle, so basically water flow, xylem and phloem. Then we have these cells here. This is called the root cortex, so it's where the cellular part of the root. And then we have these little root hairs that are coming off. Okay. Remember when we were talking about grasses and forbs that might have rhizomatous reproduction? They would reproduce by sending out roots. Those are the rhizomes, those little root hairs, rhizomes. And these here are called rhizobia. This is just the example we're giving. Those are the bacteria. Okay, and as this root hair grows, the rhizobia will infect the root hair, and the infection progresses to the cell cortex. And it makes this nodule. It makes the root hair go and get really fat, right? And it makes this incubation space for the bacteria itself. Okay, so here's the apical meristem. That just means the growing tip of the root. And it becomes this apical meristem of the nodule. Okay, so the initial infection was here. These infection threads are going this way. And then the bacteria inside here start to actually kind of differentiate. They're almost becoming like a multicellular critter. They're not, but they're starting to have some of those behaviors. And the bacteria that is in this region, this is the nitrogen fixing zone. Once the nodule is nice and big, they'll do that nitrogen fixing here using what enzyme? Nitrogenase, right? All right, and so we still have, now this this whole root cross section is smaller than the nodule itself, which is often true. So here's a picture. Here are the root nodules. They're as big as the roots themselves, and in some cases bigger. This is what we call a symbiotic mutualism. So a mutualism is a relationship between two organisms where both organisms benefit. Symbiosis, you guys probably know what symbiosis is. What's your understanding of symbiosis or symbiotic relationship? You can give me an example too. Chris.
Could pollination be an example?
Pollination can be an example of a mutualism. Absolutely, both are benefiting. I wouldn't call this symbiosis. What does it mean to be a symbiosis? Sym means same. This is a symbiosis, but the pollination example is not. So what can you think of that's different between the relationship between these two organisms that are benefiting each other and the bee and the pollinated plant? In both cases, both parties are benefiting. What's the difference between the two? Emma, you got this.
Well, pollination isn't keeping the individual plant alive.
Pollination isn't keeping the individual plant alive, it's just allowing it to reproduce. Okay, yeah, that's a good example. What are some of the other differences between those two? Sam, you know this. Give me one example. Well, okay, so where are the bacteria in relation to the plant? Inside of it, right? Are the bacteria going to do really well outside the plant? This particular species is not, right? So the symbiosis is the bacteria, one species is actually using the other species as a host, right? So we can think of negative symbiotic relationships, right? When we're infected with bacteria. This is a symbiotic mutualism though, both are benefiting. So when we think of symbiosis, we automatically go to something that's good for both parties. That's not true. Symbiosis just means one organism is using the other as a host. Okay? And if it's mutualistic, then both are benefiting.
Think about it in terms of the intimacy of the relationship between these two species.
Right, so if the bees actually lived inside the plants always and needed that in order to survive, then it would be a symbiosis. All right, I want you to think about what both partners are getting out of that symbiotic mutualism. And when you come in to class on Friday, we're going to finish up the fluxes. We just talked about the input here of nitrogen to plant, well, to microbes that are in the plant roots. And so we'll continue this drawing on Friday and finish it on that day.
I do that very